So on behalf of the Gold Country Grove Society, it is my honor to introduce Marlene, the plant lady.
That's me. Technically, my real job is as a horticulturalist at the UC Davis Botanical Conservatory. And some of you may not know that. Some of you know the Arboretum. And we house rare and tropical plants. We're known for our corpse plant, the stinky plant. We were one of the first institutions to get it blooming. We have carnivorous plants, rare succulents, tropical plants. So we have over 4,000 different species of plants, some of them in outdoor beds, but a lot in potted plants. So that's what I do. I've been there 11 and a half years. I have my degree in horticulture from UC Davis, but I've been gardening since I was 6. I interned at Sunset Western Magazine, their test gardens, and the Winchester Mystery House. And one day I got a little upset that everyone thought the conservatory was the Arboretum. So I contacted a local TV show, and then I said, hey, you guys need a garden set. And so that's how I got on Good Day Sacramento. But I also love gardening, obviously, so those roses are from my former garden. I'm particular about my roses. I like just the old English roses, David Austin roses, and I had over, I think, 80 roses at my last house. Okay, so if you guys are into social media or whatever, there's my info right there. That's my email, mrsimon@ucdavis.edu. And I write for the Sacramento Bee every other Saturday. I have an article in there. And then I also do a podcast, Flower Power Garden Hour. And it just so happens that I did one half of a rose episode, and I've yet to do the second half. And I would love to have one of you guys be a guest. So if you're interested in being a podcast, you can wear your pajamas. It's really casual. It's not live, so if you drop the F-bomb, it's totally fine. Actually, though, we leave the F-bomb in because, let's be honest, us gardeners, we really need to try to act a little bit cooler, So if F-bombs do that, then so be it. So fungus? Oh, yes. I like it. Yes. And that's worse, actually. That's worse than the F word. It's fungus, right? Yeah. Okay, so that's that. But, okay, today we are going to talk about plant hormones and how plants grow, really. And I'm going to focus on two major hormones, because when you're pruning your roses or any other plants or growing, those are the two that you really have to really sort of know. I mean, you could grow plants, but personally, I like to know why a plant is doing what it's doing. And this helps you become a better gardener. Some people may know all this. If you do, just pretend you don't. Humor me, right? Okay. So here, we're going to go through some of the parts of a plant first. Right. So that is the shoot apical meristem. We're good, right? It's where the meristem's at, top of the part. So think of a redwood tree. I say redwood trees are very apically dominant. You never want to cut the top of your redwood tree off, because then structure changes and it goes a little crazy, right? So that's the shoot apical meristem. And then your roots. Your roots have meristematic tissue as well. And between where those leaves come out, those are the nodes, right? Right there. And that's where your axillary bud is at. And a node is where the stem leaf is connected. And that is a very important part of your plant, right? That is where that meristematic tissue is at. And I'll probably talk faster than the slides go. Actually, maybe I'll catch the slides up. I'm just going to put all this there because that's what I want to talk about. The meristematic tissue. That is the area of undifferentiated cells, right? At that point, depending on what you do to the plant or the environment or what happens to the plant, those cells could become anything they want to be. When you're a kid, your parents are like, you could be anything you want to be. Actually, my parents didn't. They're like, no, you can't. But that is why the meristematic tissue is so important. And that's why technically stems are the most important part of the plant. You may find plants that don't have roots, such as tillandsias. You may find plants without, leaves. But you'll never find a plant without a stem because of that meristematic tissue, because that is what ensures the plant continues to live. Whether something eats it, something breaks, or you prune it off, that ensures. So that's why those nodes are very important, because they house the axillary buds, the apical bud, where that meristematic tissue is at. So it's basically insurance, right? So there's your inner node too. Those are good to know, right? Because if you're doing a cutting. So your two major hormones, auxin, and I'm just going to put these on here. I'll let you guys read ahead. If you're like me, you're like, okay, next slide, next slide. Okay, auxin. So auxin is one of the major hormones because it does multiple factors. So it's produced in the actively growing young leaves and shoots, and it moves with the phloem transport, the sugar transport of the plant. And it's inhibiting all the side growth. So it is actually forcing the plant to be more like a redwood tree and growing straight up. And if you use rooting hormone, it's actually a synthetic form of auxin. And if you've used 2,4-D herbicide, which no one's going to admit to it. Nowadays, people don't even want to admit they use Roundup, though. No, not me. So no. It's actually a synthetic form of auxin. So really, hormones in large concentrations can do damage, right? Just like with people. You know, more hormones aren't better. Just look at teenagers, right? All right. So cytokinin is produced at the roots. Now it moves in the water transport part of the plant, the xylem, and it's promoting that side growth. So you notice you have hormones, one moving down, another moving up. One's preventing side growth and the other one wants to do side growth. So they're sort of competing against each other, but in general, that keeps the plant in check, right? So else you just have plant growth everywhere. And if you've ever used kiki paste, that is a cytokinin. Anyone grow orchids? Yeah, you know those little plantlets that grow off the plant? They're called kikis. So the kiki paste is derived from that because you have this flower stem and you're like, well, why is it growing a plant now? Well, it got a little extra boost of cytokinin and it's producing side growth. So if you want to sort of manipulate plants, you could get some kiki paste and do it. And I'll show you down the road on here where we've done that. And I'll only show the successes, not the failures. So I'm just going to skip that one. Okay, so this was Darwin. Some of you may have heard of him. So he did an experiment where auxin and plant growth. And really what I just want to get from this is that auxin moves with gravity and it's phototropic, meaning it moves away from the light. So just keep that in mind. So you have this hormone growing at the tips, formed at the tips. It's moving downwards, gravity. It's moving towards the dark. Now we're gonna all put this into action when we're doing cuttings, but starting to make sense, right? So if you think about it that way, when you go out to your garden and you see how plants are growing, you might think, okay, this is what's coming into play. So what happens when you prune plant parts? Now we do a lot of pruning at the conservatory, mainly because our ceilings are only about 20 feet tall. So we have these tropical tall trees, and unfortunately we have to prune them a lot. And there's only so much space, and so we have to prune them a lot. So what happens when you prune a plant? So there's our plant. You're gonna make a cut right there, right above that node, right? And then you're gonna so when you pinch a plant, when you hear the term pinching a plant to make it bushier, that's what you're doing. So when you have less auxin, more cytokinin, you're going to get shoots, right? So when you pinch that plant, you're cutting off that auxin that's going down and inhibiting that side growth. You're going to increase the cytokinin at that spot and allow it to take over, and therefore you're going to have new growth. So that's what's going on when you're pruning a plant, is you're messing with the hormones. It's sort of fun. Shows how in control you have of your garden. And then the end of summer takes over and you realize you have no control of your garden. If you were to see my garden, oh my goodness. So, all right, so then, see down, I'm gonna put the little, right there. I cut a root. All right, anyone ever have a tree where you cut a root and it starts growing? Yeah, because you've just forced that, you pulled up that cytokinin, right? That's moving upwards, and you cut off the auxin moving down, and it's doing the same thing. So sometimes you remove a tree, but there's still a piece of root in the ground, and there's enough hormones and moisture and everything that then it will start sprouting. You're like, what's going on here? Well, this is what's going on. Hormones is what's going on. And then we have another one here. This is exactly what you do when you make a cutting. Cut right below a node, and then of course, let's see where we go. You're gonna have roots forming there, and then of course anywhere you cut, right? Because you've just forced auxin now to pool right there, and auxin's the root forming hormone. So when you cut above, you're gonna get some growth, but then when you make a cutting, you're pooling that auxin, has no other place to go, and it's gonna form roots. So that is what you're doing when you're making a cutting. So those are the two major hormones that are coming into play. So right there, when you increase auxin, you do decrease cytokinin, what's gonna happen? What are you gonna get? Roots. Remember, you get 1 credit for this, so there will be a test at the end. So, all right, so this is a sad plant from work. Okay, I'm moving away from, there we go, moving this way. Okay. Okay, so this plant was getting a little too big, and it's outgrowing its pot. Now, this goes for roses too. This goes for pretty much anything. It used to be at the conservatory, we would prune everything and then transplant everything. And then we would wait and wait for growth. So now we go and we prune, and this is not the best pruning job. Just ignore that part. But it was one of the plants that was growing out of its pruning. Now there's new growth happening. Right now, the auxin is at its peak, right? Because auxin is formed at new growth points. So that auxin is at its peak. It's moving down. It wants to form roots. So now if we were to transplant this, it will form roots better than if we had pruned it, took off all of its root forming hormone, then disturbed its roots, put it in new soil. It's not going to form roots, right? It would take a while. So sometimes you see a plant go into this limbo stage when you transplant it, where it's just sitting there. So now we prune, we wait for them to grow a little bit, and then we transplant. Or we prune, we forget, and it grows back completely the same size, and we have to prune again, go, oh, we meant to transplant that. So, of course, some plants we know are going to be fine no matter what. But for, plants that are difficult to grow, plants that are really big, this is a good thing to keep in mind. This is why when I tell people, they're like, oh, I want to transplant this plant in my yard. I'm like, okay, here's what you're going to do. You're going to prune it down. You're going to wait for new growth. Then you're going to transplant it. And I know some people, even I've seen this method where you sort of dig up the root ball a little bit, one side, and then you leave it, because now new roots are forming, and of course that's more hormone production going to the shoots. You come back on the other side, you dig that side up, new roots are forming, and then you dig up the whole thing and move it. So you're just playing with a different set of hormones. For me, it's much easier, and for us, because most of our stuff's, almost all of our stuff's in pots, is to prune it and then wait. But, if you have a tree and you could wait like 6 weeks, 2 weeks on one side, 2 weeks on the other side, and then dig it up. So that, if it's a special plant, all of our plants are special, but you know what I mean. All right, so why should you bury a tomato plant deeper? How many of you grow vegetables? Yeah. Was it a good tomato year? No. Yes. That's exactly what I've been hearing. Yes, it was a great year. No, it was a bad year. How many of your tomatoes took a long time to ripen? Yeah, we had a weird spring. It was, yeah. Temperatures were all over the place. Now, how many of you have tons of tomatoes? Yeah. Yep, exactly. Okay, sorry. Digress. Okay, so here's a normal plant, right? What happens when you get a tomato plant? You've heard this, right? To bury it, you remove all the leaves, and then you're going to bury it. Yeah, you get more roots, right? A bigger root system faster equals a bigger plant faster. More cytokinin, which is produced, right? And then it's going to grow faster, right? So you get more roots, more cytokinin going up, gives you more growth faster. There's also the burying method, the trenching method. And I had, anyone know wild boar tomatoes? Brad Gates. No one grows the wild boar tomatoes. Yeah, do you know him? Yeah. So he brought up a good reason why he does prefer to bend the stem and bury it. He says it's because early on, remember, you don't want to plant your tomatoes until temperatures warm up. So if you're digging deeper into the soil to bury your tomato, there could be a few temperature gradients. So by just building a longer like trench and burying it, it's warmer. So I'm like, smart. But, you know, I'm usually late planting my tomatoes, so the soil's fine. All right, anyone know what this plant is? Yeah, cactus, yeah. This is a jumping cholla. This has a really interesting, they get about yay tall in the southeast, and they have an interesting vegetative propagation. So they rarely flower, and as you know, when cacti flower, they have a very short-lived flower. So, you know, the chance of being pollinated, you got to put all your eggs in one basket, have a big, beautiful flower, but this one rarely flowers. So what it relies on is pieces breaking off. That was my hand. And that's a student laughing. So what happened was I went to go try to get that off, and I had both hands stuck. I'm like, this is great. So I couldn't even open doors. So, so the spines on a cacti are actually the leaves, right? On a cacti, the spines are leaves. Not on all succulents. Other succulents, they're modified stems or epidermal outgrowths, but on a cacti, they're the leaves. So where do those spines grow out of? What area? The stems with the nodes. And at the nodes is that meristematic tissue. So this plant basically forms its own cuttings. So this would stick to an animal, walk a few miles, lay down, roll around, it would fall off. And once that side hit the ground, there's that meristematic tissue. You got gravity pulling it down. It's touching the ground, so it's not getting any light, and it's dark, and voila, it just starts growing a new plant. So this is, don't be upset when if you Google jumping cholla, I should put this in there. I really feel bad for the guy. The second picture that pops up is a golfer covered head to toe in like foot-long pieces of the jumping cholla. He was in a golf course, probably in the Southwest, and fell back. And then what I love is the paramedics and the police standing around like this. I don't know what to do. And the third picture on Google is him on a stretcher like this. Yeah. I don't know what the first picture is when you Google jumping cholla. Probably a jumping cholla, I would hope. But yeah, that guy's for years, he's been number 2 when you Google. Poor guy.
Marlene?
Yes.
Try running to a jumping cholla at 3 o'clock in the morning while collecting bugs in the middle of the night.
No, let's be honest. How much tequila did you have? You were it was you're telling a friend's story. Gotcha. That would not have been good.
Oh, it was very painful.
Yeah. Oh my gosh, I can't even imagine. You didn't get any infections though, right?
No.
Okay. Yeah, because these things they're hooked. They're like fish hooks. They go in. I had a big, not one of these, but another cactus on my shoulder, and I backed into it at work, and I told the student, I said, oh, can you get this out? And I thought they were going to do it gingerly. I'm like, well, that actually was like ripping off a Band-Aid, so it worked. See what else? Okay, gotta go back there. All right, so at the conservatory, we do a lot of cuttings. If we want seed, we either hand pollinate, or every once in a while, if it's self-pollinating plant, it'll form fruit on its own. But for the most part, all the plants that we propagate, we do from cuttings. So we have a mist bench, and we do cutting. Some root very easy, others do not, and sometimes they take years to root. There's this, I'll show you a plant that is my nemesis. But here's our mist bench. We also have something called Easy Clones. This is a company, and they've donated these to us. They're like a hydroponics unit. Some of our students are like, God, I really want one. I'm like, you could build one really easy. Just, they started with the cannabis industry. right? So it's just a container with a little micro sprayer. So the idea is a lot of air circulation and some moisture. And then they built these where there's lights. So you have the light. They work really well. I'm not a huge fan. I shouldn't say that. The company is great. Everyone go out and buy one. Just kidding, because they've donated all these to us. But this is another way. So some things actually that we can't get rooted in our standard ways get rooted in these. And then vice versa. Some things just don't take in here. Okay, so when you're going to go do a cutting, usually you want the new growth, right? The newest growth has the most auxin. So you hear sometimes of doing woody cuttings. Well, remember when something is more woody, it has that less auxin in it, so it's going to take longer to produce roots. So you could see just right here the two differences. So this is a Poliscius, a houseplant. So even right here, you could see, let's see, that this one's younger, closer internodes, so you know that one was a younger piece, and this was an older piece. And the root, same time, just better root formation. Big difference there. You know, this isn't a very difficult to root plant, very herbaceous, but even that, an herbaceous plant, same plant, the new growth versus older growth, you could see how much difference there was in the auxin, which equals quicker root formation. And with that, we have a whole assortment of rooting hormones at work. Now, this is the one that you usually get at the store, right? Something like that has a little bit of a fungicide in it, which sometimes that's worth it. But then we have the Hormex that goes from 1 to 45. And yeah, we've got some good hormones at work, yeah. So with a woody hibiscus, I would use like a 30. I don't know if we've ever done 45. But something woody, we would use 45 because you need more auxin, right? You're trying to manipulate a woody part that really doesn't want to root, so you need to go stronger. And a little bit goes a long ways. We hardly ever use rooting hormones now. When we do our cuttings, we make sure it is actively growing tissue, and we don't even need to use the hormones. Now, some people will say, well, things rot out really easy. You could use a little bit of dusting sulfur on the cutting to just act as a natural fungicide. Believe it or not, honey is antimicrobial. Cinnamon, salicylic acid, like willow bark. I don't know, I've always wondered if like Bayer aspirin, if you crush up a Bayer aspirin, because that's true aspirin, and dipped it in there, if that would work. But all those are going to be antifungal. So you could, if you were like, oh, I don't want to use a rooting hormone, but I'm worried about fungus, you could dip them in there. But also if you just use a clean media and don't keep do it properly, you're going to have some death, right? I imagine you guys, when you do your cuttings of roses or anything, you make sure you do a few extra, right? Yeah. Yes?
Do you have a preferred media?
We'll get there.
Okay.
You bet I do. So, yes.
I heard that rooting hormone after a year is ineffective. Is that true?
No, that would be great marketing. But you don't want it to get wet, and you don't want it to sit out in the sunlight because that breaks down anything. And you do want to wear gloves. It is, like I said, a strong auxin is an herbicide, so you don't want this. I'm not a plant yet. I was hoping enough auxin I turned into a plant, but no, it doesn't. These bottles we've had for years. Good point. I don't know if they really work. I mean, maybe they last for years. Just don't get them wet or in the sun. Okay, so I just wanted to show this, what happens so here's a cutting, right? With the leaves, forms a callus, and then forms callus into roots. You take the cutting, you remove the leaves, and you add the synthetic auxin, and it could still form roots. I don't recommend doing that. Sometimes when you're doing a cutting, you don't have a lot of leaf material, so you feel like you're almost removing all the leaves. So in a case like that, yeah, you may want to use a rooting hormone. If it's, say, a tropical plant or has big leaves and the internodes are very long, and you're like, wow, I have like one leaf, maybe I should add some auxin. But also think about the stem. The stems are also photosynthetic when they're green, right? Even when they're brown, there's still chlorophyll and they can still photosynthesize, but if the stem is green, it still is photosynthesizing. Not as efficient as leaves, right? But it's still doing that for you. Okay, so here's our media of choice. For years and years, we would use 50% vermiculite, 50% perlite, the small perlite. We found on a mist bench that would create enough aeration, but then the vermiculite held enough moisture. We also use straight perlite. I find that that is just as good as the vermiculite and perlite, really. So you could see here's some of our pots. And you know, I'm sure you all know when you do cuttings, you could put multiples in a pot because they're not competing for root space. It's more about how much air around the leaves they're getting and how much sunlight they're getting. So we have multiple cuttings in one pot. We use short pots, ideally, because there's no roots right there, right? So shorter and wider. These are begonia leaves, and I'll show you in a bit what we're doing. And we have a heat mat. A little bit of heat mat helps just to warm up, and of course when the roots start and there's callus, the warmth is going to help that just a little bit. And of course it's on a mist, so the moisture, we have one of those, it's really still like the simplest, nothing fancy, those leaves. You guys know what I'm talking about with those leaves? When the moisture gets on the metal paddle, it goes down. And then when it's dry, it goes up. It triggers the misters to go on for 15 seconds. So it just regulates the moisture. So it works for us. And I'll go through some more media in a bit. Okay, so leaf cuttings. Anyone do leaf cuttings? Yeah, so this is an African violet. So remember I was saying stems are the most important part of the plant because they have meristematic tissue? There are a few plants that their leaves have meristematic tissue. And with that, you can form new plants. So there's exceptions to everything. And some of them are, the African violet, Sansevieria. You guys know the Sansevieria, the mother-in-law's tongue? Yeah. I always get in trouble when I'm like, because sometimes mother-in-law's tongues could be a little sharp. That gets its name. Let's see what else. There's sedums, echeverias. Hydrangeas, you need stem cutting. Yeah. Some of the, let's see. I'm drawing a blank, but I'll show you. There we go. Okay, begonias. There we go. That's why I have there. That's iron cross begonia. And it's very pretty. So a lot of begonias, you could just do stem cuttings, like the cane begonias, you could do stem cuttings. Others, if they're Rex begonias, they don't have stems, and you want to make more of them. So you could do one leaf, or you could do the confetti method, where you take a tray with perlite or your vermiculite perlite, and you cut up a leaf into multiple sections. Now, what we find is if there's a major vein in the cutting, it takes better than say if it's off on the side and it doesn't have like a major vein. Good thing that begonias are, have a lot of venation. So I just lay this on top of the mist bench. I try to make contact, but no need to really bury it. And then you'll have little plants forming. So out of one leaf of the begonia, I got 10 plants. One leaf, 10 plants. And you can say, well, look how many pieces you had on there. But still, you're going to have death. Death happens, right? So even with a regular cutting. So that's just one. Not everything can you do that with. Like the African violet, you're going to need that little base where the petiole attaches, right? Because that's where your meristematic tissue is going to be. So it just happens with begonias, you could even take multiples. The Sansevieria, you could take cuttings of that Sansevieria leaf and root those into individual. Now for all of our succulents that we do cuttings of, we use pumice or red lava, right? And for those you want to let callus over, when you do a succulent cutting, you never want to stick it directly into the media. You want to let it callus over in the air, because if you stick it in a media, even a dry media, it could rot. That's why we recommend that when you transplant succulents, you never water them in right away. You wait a few days because they're very susceptible. So for all of our cuttings, we use a pumice, red lava, and we don't put them on the mist bench. We'll put them on a bottom heat bench, but then we'll water them just very sporadically. And then they root, and then we move them up into soil. So that's a major difference for your succulents. Any questions?
You do the same thing for a Opuntia cactus?
Well, I wouldn't cut it up. But yeah, I mean, everything on a puntia is a stem. So if you cut it, all those spines are meristematic tissues. I don't know what happened if you would lay it on its side.
It roots.
Yeah, that's my guessing. It would root. Yeah. It's probably why when you see clusters of them, you see other, because they fall off, they land on the ground and it starts rooting. So yeah, it's the same idea. You got your node, you got your meristematic tissue, and then you're going to form roots. Okay, so just ignore the hokey me. It's so this is what I wanted to focus on. So when you're in the plant world for so many years, you miss trends. I did not know that houseplants were it and cool and hip until about like a year ago, right? And I did not know the fiddle leaf fig, this one right here, Ficus lyrata, was like an it plant. When I found out, I almost died, right? And my joke is, hey, the 70s called, they want their plant back. I mean, right? I mean, these were just, you see them in office buildings, and now they're just like crazy expensive. The Hoya, anyone know Hoya?
Yeah.
Yes, beautiful, right? So there's one called Hoya caryi. It's a heart-shaped leaf. Most Hoyas, you could do stem cuttings. They root relatively easy because they have adventitious roots all along the stems anyways, like a lot of tropical plants do. But the Hoya caryi leaf, you could bury and it will form roots. It will almost never form into a plant. It has the material to form adventitious roots, but it doesn't have that meristematic tissue to push out a new plant. So they sell that. They actually sell just leaves that are root. I'm like, why would you want that? Confused. So I know with the ficus you could do that. And then someone told me, oh, no, I got plants out of it. So of course I had to go, I'm gonna go in. Our poor ficus was in the greenhouse for years. We're like, oh, we have no space for this. Let's just throw it outside, give it away. And it ended up in our hallway for years. And then next thing we know, it's like the it plant. So we're now propagating off of it. And I know, I'm like, it's in our hallway, Ernesto. I don't know, what should we do? Throw it away? No, let's chop it up and sell it. Yay. So I took 2 leaves and did the same thing, vermiculite and perlite, and buried them. And sure enough, they formed roots. They've been sitting there for probably about 8 months now. They're not going to form plants. Same thing. It has enough material to form those adventitious roots. There we go. But it's not going to form. So maybe 1 in 100 might, but I'm not going to do 100 cuttings of that because I know we could do a stem cutting and it'll root right away in water pretty much. So, certain things that are leaves will be able to form roots, but they can't form into a plant. So it runs the gradient of where the hormones are at. Let's see. All right, here's my nemesis. Yeah. So this plant is called a needham, and it is it's actually related in the same order as the welwitschia, which is a strange plant unto itself, but it's an ancestral plant. It's a coning plant, but it looks like a flowering plant, so it confused taxonomists early on. Those taxonomists, they love to change names, right? We got to find new jobs for them. So my memory is not like it used to be. When they changed plant names, forget it. It's still the original when I learned it. Okay, so this plant, I had a cutting formed in one of the easy clones. I took a fresh cutting. I tried all different kinds. I even tried air layering, which I'll go through. And it sat in that easy clone for a year, and it would callus over. It would even put on new growth. Have you guys ever seen that where you have a cutting? It puts on new growth, and you're like, for sure there's going to be roots. Nope, no roots. And so I left it, and one day there was a root. I was so excited. So I potted it up, and because that's what you do, of course, after it forms roots, and I put it off to the side. And I went to go pick it up one day, but the leaf it had was stuck under another pot or a lid, and it ripped. The whole top part ripped off of the base of the roots. You could hear me screaming, I think, through the whole entire campus. I mean, I was so upset. Since then, we've been able to root others, so that's good. But this is something bizarre that this plant does. This is a leaf. it's growing new plantlets on its leaf. Those are all plantlets. Now I took that and I tried to get it to root, didn't happen. So here, there was probably more, what would you say, possibly in there? Yeah, I would think cytokinin, right? No roots, so it wouldn't really be auxin. There's foliage, so it's possible that leaf has a little bit of cytokinin. You would think, okay, well, there's cytokinin forcing foliage, so then there should be some auxin right there that would go down to form roots, but it didn't happen. So yeah, it just rotted out. So I'll probably try again, but sometimes hormones get very confused. Not saying anything. So, all right, anyone know what this plant is? Looks like an avocado, yeah. It's not. That's one of our corpse plants. So our first corpse plant was, so I know you're like, oh yeah, Sac State has them, Chico has them. They got them from us. Years ago, I think when I was a student at UC Davis, we got some seeds from like Borneo area, and we grew them, and they take years to even get mature enough to flower. And then the first one we had flowered, we nicknamed Ted. Now, apparently, this doesn't get out of this room, okay? Apparently, it was nicknamed after the stinky chancellor we formerly had of that name. I don't know who that was. Yeah. Oh yeah, it was Ted. So, interesting thing, if you know the corpse plant, even if you have the jercunculus in your garden, some of the voodoo lilies, it's the same idea. They grow from a corm. Now, remember, a corm is a modified stem. It's a very compressed stem, and like a potato, right? And the eyes of the potato are your nodes, right? So when you cut up, if you grow potatoes, you can cut those up and bury them, and you can have new potatoes as long as they don't rot out. So when you have one major stem and it's very compressed and animals want to eat some of them, you better be buried. So the corpse plant has one apically dominant node. And so it sends out this giant leaf, and the leaf gets about 20 feet tall. It looks like a stem with leaves, but in fact, it's only one leaf. It dies down after a year. And so it's, besides seeds, it's very difficult to propagate, right? And it's really hard to set seed. We had one that bloomed, and it's not one flower. It's an inflorescence. It's made up of hundreds of little flowers. Half of it so think of like a calla lily. So half of it got pollinated. Well, because it took so much energy to form all those seeds, the plant died. So we don't want ours to be pollinated. We want to continue it to bloom, but something pollinated, probably some fly. So how do we make new ones when it doesn't really offshoot? The big corpse plant doesn't send offshoots off of its corm. And you have one leaf, you have a stem that you can't really cut up. So we actually can do leaf cuttings, portions of the leaf of it. So this is another one where a portion of the leaf, we use rooting hormone for this one. And so we just take part of the leaflet, dip it into root toe. Just remember, a little bit goes a long way. And then we stick them on the mist bench. And there they are. There's our cuttings. And we use the vermiculite perlite for that. I know some people use sand. Sand is really heavy, and it holds a lot more water than most people realize. They think, oh, sand, it's going to be air. Perlite's much better way to go. So there's so these we sell like $100 a piece. We'll talk later if anyone wants one. Okay, so I just functions of cytokinins. Okay, this whole list here we'll just go over real quick. Stimulates the growth of lateral buds, release of apical dominance, right? So you have an apically dominant, it's going to release that. Allows leaf expansion and enhance stomata open. You guys know what stomata is? Yes. Okay. If no one said no, I was going to have to say, well, what's stomata with you? Sorry, that's a bad pun we use all the time. I stole it. I'm sure Ernesto stole it. So stomates are important because that's how the plant does photosynthesis. And I was mentioning that stems are incredibly important and they could do photosynthesis, but leaves have more stomata. So that's why they put on leaves, right? And that's why you see desert plants that don't have leaves, because you're going to lose water through. So stomates are important. So that's cytokinin. And this is what I was talking about, those kikis on the orchids. So sometimes it'll just, you have a flower, so say you get a phalaenopsis and it's done flowering and you leave it on there, all of a sudden there's a new plant. And I get questions like this all the time. First of all, I get questions, my orchid stopped flowering, is it dead? I write back very nicely. No, it's not, but sometimes they'll send new plantlets off, and those are called kikis, and you could break them off, and then they form roots because there's cytokinin action going on there. So, and what I want to show you is some fun with kiki paste, right? So here is a cactus. And so if you picture it having like a dome being one cactus, well, we wanted more of this one, and it wasn't flowering. And we could chop it, right? You could chop sections off. So you could see right there, we chopped it. And because that was a node, it's sent up. But we decided to sacrifice this and see what would happen if we took a knife, cut into the main meristematic tissue, and added that kiki paste, which is cytokinin, which promotes foliage growth, right? And sure enough, look at, not blooms, we got new little babies. Which, as you know, if you grow cacti, you could snip off, right? Let them dry and then pot them up. That would have never done that unless we added that cytokinin paste. Now, remember I was saying I was only going to show you the successful ones? We killed many plants doing this. Because what happens when you cut into a succulent? Yeah, I mean, that's like I always joke that's like cutting open your arm and going, I think I'm going to pack it with dirt. That's a good idea. So that's why you never want to water succulents right in, and you want to let them callus over before you put them in your rooting media, because they, it's just like a big open arrow pointing like pathogens, fungus this way. So we ruined a lot of them. And I don't recommend using a serrated knife. Use a sharp knife, because a serration is gonna, just gouge everything. But this is the kiki paste. And so if you have a plant that isn't forming seeds, you can't get to bloom, you can't really do cuttings of, but you think you could get little plantlets off, you could have fun with it. So, all right, anyone try air layering before? Yeah. So air layering is the idea that you're making a cutting while it's on a plant still. They do a lot of tropical plants. The one that we like to do and have fun with is the Ficus lorata, because it's our sacrificial plant. I did try the Needham, my nemesis, and it didn't work. So the idea is that you get a woody, you can play around with it, but the idea is a semi-woody portion of the plant, and you just sort of scrape that cambium layer off. You want to do it directly below a node, right? As like you were making a cutting. And you can add hormones, or if it's fairly new cutting and you think there's enough auxin forming, you don't need to add hormones. We then put wet sphagnum, and then you put plastic around it, and you tighten it really tight, and then you put foil over it, and so the sun doesn't dry it out, and you leave it on there, and you could peek, and it'll form roots, and then you cut right off below it. So you could do big sections of plants and do instant cuttings without having to misbench or without having to worry about your cutting rotting off. Now, you have to, sometimes you could cut too deep into the plant. I use just a straight razor blade. If you cut too deep into it, sometimes you're like, oops, no one saw that. Cut that off and then move on to the next, So in certain plants, it works really well for, like I said, a lot of tropical fast growing plants do better. And I wouldn't do very, very fine new growth. It's just, there's no cambium woody layer to really cut into. So, but it's fun. You could definitely try it on roses. Do it on roses. And then there's just sort of the cambium layer. Yeah, I don't know if you guys seen, the cambium layer. And then you have the phloem to the outside and the xylem to the inside. So you're just sort of scraping off of that to allow, basically scrape off the bark in the cambium layer. All right. I'm sure this has happened to all of you, even on roses. What if this is my last slide? Ooh, no, I have a few more. Don't you worry. Am I good on time? Okay, excellent. So I'm sure you've gone out and you're like, oh, that branch fell off, and you go to pull it off, and sure enough, it's rooted, right? So it's the idea is trench. Another way we do cuttings of things that are difficult, or you could do at home if you don't want to cut the cutting off of the plant, is you could do trench layering, which is basically taking a bendable, so that's the key part, bendable. So it has to be, new enough that you could bend it. You could either do a pot with either a direct soil even, or a rooting media, or you could do it to the ground and pin this. So remember, when you're doing a cutting, you need at least one node below the planting media and at least one node above, right? So here I'm bearing 2 nodes. Ideally, I would remove these leaves here. I would pin this, and then you have all this as your top growth, right? And the idea is that's going to form roots because auxin is moving with gravity. It's moving away from the light. It's pooling right there. And so roots are going to form. And when it forms roots, you simply just remove it from the mother plant and you have a new cutting. Now, remember I was saying with how just now the hormone moves with gravity? So when we do our cuttings, especially our succulent cuttings that have to callus, do we lay them like this or do we lay them like this for them to callus? This way. Right? If you lay them on its side and you're talking like weeks to months, all that auxin is going to pool on one side. And when you go to plant it, you're going to have one side more dominant and possibly roots forming faster on one side. So you want to leave them upright. And of course, a dry spot. Every once in a while, we have this tall cacti that we want to make cuttings of. And so the top's very easy, right? Because it has the dome. You're like, oh, that's the top. But think about a tall cactus. In midsection, you cut it, there's no way to know which way's top and which way's bottom. So we'll draw arrows with a black Sharpie while they're callusing, like this way's up, because that would be really embarrassing if we planted a plant upside down. Of course, we wouldn't admit to it. We'd be like, something is wrong with this plant. It's never us, no. And that's the thing, is that we kill plants all the time, but it's never us, right? I mean, something happened, but not us. Right? Okay, so I just want to talk about a few more hormones. The ethylene. We all know ethylene, right? Because it's sort of the death hormone, right? Don't put your apples in with, or your bananas in with your apples, or do so if you want them to ripen, because it's a gas that emits. So it releases dormancy, stimulates fruit abscission, bromeliad flower. A question I get a lot is people who are gifted bromeliads. You see them at the store, right? The gift bromeliads, and they're in full bloom. And people get them and the blooms start dying down. And they're like, what can I do to get my bloom? And I'm like, you got bigger problems than your bloom. Because a bromeliad's monocarpic, most of them, meaning once it flowers, it dies. And by then you hope it has set little babies off. But what they do at nurseries is they usually pump them full of hormones to get them to bloom so they're ready for market, but the plant's not mature enough to set off little blooms. So if you want a bromeliad or gift a bromeliad, try to find it from a reputable nursery, unless it's, I actually told my little side story here is my mother-in-law was having to buy flowers for a funeral. And she's like, well, I figured I should ask you, should I buy them a plant? Like, no, I get so many questions where there's so much sentimentality in that plant that people are freaked out about killing a plant that was given to them at a funeral. I'm like, buy them flowers. They know they're going to die. That way they don't have to stress. Sorry, that's just my feeling, right? Sorry. Unless it's a rose, of course, right? But yeah. Fruit ripening. So you could just read this. I'll flip another one here. Gibberellins. Interesting story about gibberellins. So gibberellins are responsible for cell elongation. So if you have something really stretched out, that is gibberellic acid going on. So it stretches. So if you have a leggy plant, that's what's happening. But it also has to do with seed development. Anyone grow grapes? Seedless grapes? If you grow Thompson seedless grapes, how big are your grapes? Little. The clusters are big, right? Because they're seedless, they don't produce this. So if you don't have a seed, there's nothing there triggering the fruit to ripen, right? So what seedless Thompson grapes and other seedless grape growers do is they give it gibberellic acid to plump up the grapes. And I actually got my hands on some gibberellic acid. I think you could buy some now online, but I did try it. I think you're supposed to do it when the grapes are flowering, spray it once, and then do it another time, and that will plump up your grapes. Because by nature, they're like, why does the fruit need to develop? There's no seed in there. There's nothing triggering the fruit. So that's just, you could get huge clusters, but you won't get the big grapes because the gibberellic acid's not being produced there. So, something else I want to mention about seeds. Anyone know what vivipary is? You may not know it, but ever cut into a plant and the seeds are germinating in the plant already? Yes, like tomatoes could do it? Yes. So, yeah. Actually, our chocolate does it. We have a chocolate plant at work. The fruit, we'll cut open the fruit of the chocolate and some of the seeds will already be germinating. So I'm sure tropical plants, because you're not supposed to keep tropical seeds. As soon as you get a seed from a tropical plant, you need to plant it right away versus like a sort of chaparral plant. So there's a funny story about McDonald's years ago that was trying to save money, time, and they were ordering their tomatoes already sliced, but the seeds were germinating. So they gave a researcher this big grant to find out how to prevent this. Well, the joke is the researcher just simply used a different variety of tomatoes. Yeah, I was like, here, use this one. But the sad thing is he had to give part of his grant back because he finished it so fast. I'm just like, but yeah, plants. Yeah, simple as that. Just a different variety will, do different things. But that's a really interesting hormonal thing is when you cut a plant open, it could be a little like, oh my gosh, what's going on? And we have planted the chocolate seeds that have done that and they've been fine. So as long as they're not past a certain point. So I think that might be it. Are there any questions? I ended on just without even a happy slide at the end. I should have put my cat. That would be my cat picture. Yes. Oh, a couple questions. All right.
Rose propagation. One about rose propagation, one about repotting roses in the same size pot.
Okay. Rose propagation and repotting roses in the same.
Two different questions.
Two different.
First question.
Uh-huh.
When I propagate a rose by rose cutting, typically we're taught, 3 nodes above, 3 nodes below. So why is it and I've only been doing it for like a year and a half. So my experience is every time when they are ready to be potted up, my cuttings that are rooted, and so there's 3 nodes below. The roots are always at the bottom node.
Yeah. Because.
Why do I bury 3?
Could you repeat the questions, please?
Why, when she goes to repot her roses, she has a 3-node cutting down below her planting media, the bottom has the roots. And to answer that, it's because that's the lowest point of gravity, right? That's where the oxygen's pooling. Why you have 3 nodes, really, to be honest, is probably stability while you're putting your right? Yeah. Certain things will form roots from everything, but if you have like something like a rose, really, if you only buried that one, it would probably flop. So it's as simple as that. Just expect those roots to form at the bottom because that's oxygen pooling, and really it's to stabilize. Yeah.
So the rooting hormone only needs to go in the very bottom node.
Oh yeah, you only do rooting hormone at the cut point. There's no point in putting it where it's not cut. It's not getting into the plant. Yeah. Yep. And try doing it without rooting hormone if it's a new growth. Just to have a little experiment.
Okay, and the second question is, a lot of people want to keep roses in the same decorator pot, especially the miniatures.
Okay.
Or mini floras. So they get root bound.
Yeah.
And so then I've been lifting it up, kind of refreshing it, cutting off some of the roots. Well, how do you do that? When you talk about auxins versus the cytokinin, what's the best
way of doing it? So if you think of it this way.
Keep it in the
Oh
same
yeah.
pot.
Okay. So when you want to keep a rose, especially like a miniature rose, in the same pot, and she takes it out, she freshens the roots up. I imagine you mean cut some of the roots. How would you go about that hormonally? Now, what's best for the plant? So if you think about it this way, what do you do for bonsai? You cut the roots off and you prune, right? Because you're keeping it in that state of imbalance where it's not really growing. So what your goal is, if your goal is to keep your plant stunted, you could transplant it and cut those roots and put it back in the same pot. If your goal is to get the plant to grow bigger, you're going to want to prune ideally first, let it grow back, and then transplant it. And then because you're moving it into the same size pot, you are going to have to do some root pruning on that. But no matter what, when you transplant, you're damaging roots anyways. So that's the idea. So, and that goes for like when you're transplanting a rose, you wait for spring and the new flush of growth to come out. You think about doing it then versus when it's dormant. Dormant, it's in sort of its own equilibrium. But I have way better luck planting and growing, actively growing roses than I do dormant roses. I think it could be because I have really clay soils, bad drainage. So when those roots first come out, it may be a little too, even amended areas, but I only do, well, here's my rules. I only plant they're getting harder to find. My second favorite rose place, of course, David Austin's my favorite. Just kidding. But I don't do grafted roses, and I do actively growing roses. Those are my, just so you know. It's like a dating app. It's like, I like hikes. I like actively growing, non-grafted roses. So, okay. Any other questions? Those are good questions. Yeah. Why do you bury 3 notes? Really, it's stability. Yes.
What's your favorite medium for roses when you're rooting them?
Vermiculite and perlite. 100%. I've done so many roses just that way. And yeah. Mm-hmm.
Have you tried Clara?
Yeah, I've tried it. He said, have you tried core? It's mainly because what works for me with really good success. And I think all the roses I've done on my own, and I've done them on the Miss Bench at work just for other things. But I think for at work and other things, it's just so much easier to use the vermiculite and perlite than that core. But yeah, I know people have great luck with it. Don't use the potato method, please. You guys have heard that, right? Where it's you take a piece of potato and you do your cutting in a potato and then you bury the potato, and the idea is there's probably enough moisture, maybe starch, to get and then by the time the potato is rotted away, you have roots. And I do a myths talk and I have that on there, and I'm just like, well, why? People just love it. I mean, if you want to do it for fun, fine. But some of these roses you want to make cuttings of, right? I don't have time for that. So other questions? Sorry, I will just start stomping my feet now with things that's on the internet. Do I have time or am I out of time?
Well, I don't have a question, but if you are a CR, there's a list going around.
One more question and then we have to wrap it up.
Okay, one more question. Yes.
I stopped using vermiculite and just went over to perlite because the vermiculite, there was so many indications of it being carcinogenic and stuff like that. Can you fill me in as to what you know about that?
So anything we use, we wet. Those big bags of vermiculite perlite, we cut them open, we pop holes in the bottom, we have DI water and RO water, we run that through. We do not breathe any of this in. So perlite is still a volcanic glass. Vermiculite is expanded mica. Sand is silica. Anything that is of that material, even our floors, we spray down to sweep up, because if you sweep that up, all that, so yeah, and all that could cause, so even perlite is, can be just as bad. So make sure you're using this stuff wet and not breathing it in. Good point. Okay, if you have questions, I'll be sticking around. But remember, we do have a test. It's multiple choice and short answer. Just kidding.